Good morning, children. We are in the second class of surface chemistry. The same chapter, chapter 5, surface chemistry. We are discussing the second part of the chapter where we start our study with adsorption isotherm. But before that, we should know what is the meaning of adsorption isotherm. See, adsorption isotherm is a graph which is plotted against the extent of the adsorption in reference to pressure when the temperature is kept constant. So in this case, when we actually plot this type of graph, what we have seen that in this type of graph, if I am plotting the x by m in contrast to pressure, I am getting a graph like this where there is the different variation of temperature. Can you just see the three types of graphs? In this three type of graph, what I have plotted pressure against x by m. What is x by m? Extent of the adsorption which is taking place at a specific temperatures. Here you can see three temperature graph, one here, the other here, and the third one. So each of them varies according to the temperature. Okay, now if I'm plotting this type of graph, that is called as adsorption isotherm. It can also be adsorption isobar, it can also be adsorption isosphere. What is adsorption isotherm? Adsorption isotherm means when I'm plotting the extent of adsorption versus pressure at constant temperature. I can also plot it extent of adsorption in reference to temperature at constant pressure, then that is called as adsorption isosphere, right? I can also plot it against adsorption in I am plotting the temperature versus pressure with the extent of adsorption keeping it as constant, then that is called as adsorption isosphere. So here what I could see that the variation in the amount of gas adsorbed by the adsorbent with pressure at constant temperature can be expressed by means of a curve which is termed as adsorption isotherm. Now here we have the first curve, that is called Friendlich adsorption isotherm. Here we have the first curve that is called as Friendlich adsorption isotherm. Can you just see? It is called as Friendlich adsorption isotherm. So it is the relationship between the pressure at a constant temperature is called as adsorption isotherm and is given by, here you can see x by m is been plotted and here it is the pressure P. Okay, so it is the pressure P is plotted in the x-axis and x by m is plotted in the y-axis, right? Where x is the mass of the gas adsorbed and m is the absorbent of the gas. Okay, m is the amount of the adsorbent gas which is being absorbed at a particular temperature. As we are plotting it at a particular temperature, it is called as adsorption isotherm. Now in this case, when you are taking logarithm on both sides, we are getting log x by m equals to log k plus 1 by n into log p. Log k into log 1 by m in 1 into p. Now the solid first increases with the increase in pressure at a low pressure but becomes independent of the pressure at high pressure. Okay, but becomes independent at a high pressure. So here we have the slope of the line in and the intercept is equal to that of log k. Here we have the slope of the line is and the intercept will be equal to log k. So this is how we get this type of Friendlich isotherm. So just remember the expression log of x by m equal to the log of that. See this expression, this is the expression that when you take the log on both sides, that is the log of x by m equal to log k plus 1 by n into log p. This is how the expression proceeds. Now one important thing you should know, that the limitations of Friendlich adsorption isotherm. What are the limitations? The limitations are that one of the drawbacks of this Friendlich adsorption isotherm is inability to provide explanation for the condition when 1 by n equal to 0. It is unable to explain the condition when 1 by n equal to 0. That is one condition. Another condition is that when x by m equal to kp0 equal to k p to the power 0, when k p to the power 0, these are the conditions that is unable to explain, although it is well supported by the experimental results. In other words, it fails at a high pressure. So this condition is incapable at a high pressure. This much you people should know. Now the next one we have is in the slope of the line and the intercept will be equal to log k. This is a very important factor which I wanted to mention you, that the slope of the line and intercept will be equal to that of k. Okay, that is equal to k, right? See this value, the slope of the line and the intercept will be equal to that of k. This is a very important factor. After that, children, we are going to discuss about adsorption isobar. What is adsorption isobar? Adsorption isobar is actually the process of adsorption takes place at a constant pressure. So if I have a physio's option, I'm plotting x by m. I am plotting x by m. So I get this type of a graph. Or if I'm plotting a graph like this, in case of Kemi's option, the graph comes like this, that is x by m equal to t. Here I am just plotting Kemi's option, that is called adsorption isobar. Okay children, so have you understood Friend-Lewis adsorption isotherm? After that, children, we are moving to the next topic, and the next topic we are having over here is actually catalyst. Now what is a catalyst? So just remember, catalysts are those substances which alter the rate of a chemical reaction without itself getting disturbed. So catalysts, these are substances which alter the rate of a chemical reaction and themselves remains chemically unaltered, unchanged after the reaction has taken place, after the reaction has completed. That is called as catalyst. So catalysts are those chemical substances which increases or decreases the rate of a chemical reaction without itself remains unchanged. So end of the reaction, it remains actually constant. After that, we have the next one. See, just try to understand promoters. What are promoters? Promoters are actually called catalysts of catalyst. So these are the substances which increases the activity of the catalyst. That is example, molybdenum is a promoter where iron is a catalyst in the Haber's process. You know that in the Haber's process, actually iron is used as a catalyst, but the problem is that this iron becomes inactive when it is been made or when it is been intercepted into the reaction. As it becomes inactive, so what we have to use? We have to use a substance called promoter. What is a promoter? Promoter is actually a catalyst of a catalyst. What is the meaning of that? It is actually a catalyst which activates a catalyst. So in this case, in Haber's process, we have iron. Iron is the catalyst, but in this process, what is happening? Iron is activated by that of the molybdenum catalyst. Okay, iron is getting activated by that of the molybdenum catalyst. Okay, and it is always in the Haber's process. It is always in the Haber's process. After that, see catalyst poisoner, which is called as inhibitor. What is called a catalyst poisoner inhibitor? These are the substances which decreases the activity of the catalyst. These are the substances which decreases the activity of a catalyst. Examples: the arsenic acid acts as a catalyst. Arsenic acid acts as a catalytic poison in the manufacture of sulfuric acid. See, as a promoter activates the catalyst, inhibitor actually depromotes or deactivates the catalyst. See, so it is called in a way catalyst poison. So it is called in a way catalyst poison. So what is the function of it? The function of it is to destroy the activity of the catalyst, and that's why, see, these substances decreases the activity of the catalyst. Example, arsenic actually acts as a catalytic poison in the manufacture of sulfuric acid by the contact process. So if arsenic is present, then that will deactivate catalyst. Now what are the different types of catalysis? The types of catalysis are homogeneous catalysis and heterogeneous catalysis. What is the homogeneous catalyst? When the catalyst and the reactants are in the same phase, then it is called as homogeneous catalyst. For example, suppose we know that the catalyst, the reactants and the products, all of them are in the gaseous phase and the catalyst is also in the gaseous phase, then that is called as homogeneous catalyst. For example, in the contact process for the oxidation of sulfur dioxide to oxygen, if I am adding nitric oxide, so see, sulfur dioxide is gaseous, oxygen is gaseous, we are getting sulfur trioxide, that is also gaseous, but we are actually using nitric oxide as the catalyst. As all these three are in the gaseous state, this type of catalyst is actually called as homogeneous catalysis. That is, when the reactant products and the catalyst are in the same phase, this is called as homogeneous catalyst. Now after that we would be discussing next part, that is called heterogeneous catalyst. What is heterogeneous catalyst? When the catalyst and the reactants are in different phase, it is called as heterogeneous catalyst. For example, in the contact process, suppose I have sulfur dioxide, oxygen, and on oxidation it again changes to sulfur trioxide, but we are using a catalyst like vanadium pentoxide, V2O5. So vanadium pentoxide is a solid, the reactants and the products are gaseous, then this type of catalyst is called as heterogeneous catalysis. Okay, this type of catalysis is called as heterogeneous catalysis. Okay. After that, we are going to discuss activity of a catalyst. It is the ability of a catalyst to increase the rate of a chemical reaction. It is the capacity by which a catalyst increases the rate of the reaction. Next important part we have is the selectivity of the catalyst. It is the ability of the catalyst to direct a reaction to yield a particular product. That is, catalyst is very much specific for a reaction. For a specific reaction, specific catalyst is used. If I change the catalyst, obviously the reaction will not show the catalytic action, or the reaction will not proceed accordingly. After that, we have the next part is called the shape selective catalyst. Next part we are having is the shape selective catalyst. In that portion, what we know, that the shape of a catalyst determines a reaction. Shape of a catalyst determines the reaction, and that is called as the shape selective catalyst. That is, it is the catalysis process in which we see, as you could just see, it is the catalysis which depends upon the pore structure of the catalyst and molecular size of the reactants as well as the product. Example, zeolites have specific shape and they are selective catalyzed. This is due to their honeycomb structure. Due to the honeycomb structure, they have this type of shape selective catalysis. So just try to understand. The most important example is that the catalytic action depends upon the pore structure of the catalyst. If the pore structure is proper, then obviously the catalyzed reaction take place faster. If the pore size is of not that type, the catalyst reaction not take place in that form. Or we can say that zeolites exhibit a type of shape selective catalysis because of their honeycomb-like structure, which consists of truncated octahedra as their building blocks. And accordingly what happened, the reaction proceeds. After that, we have the next one is enzyme catalyst, or what are called as enzymes. See, just remember, enzymes are called as biological catalysts. That is, these are catalysts which are of biological origin and they actually enhance biological reactions. Okay, so enzymes are the complex proteinaceous substances produced by living bodies such as plants and animals. I'm repeating the definition of once again, that is, enzymes are the complex proteinaceous substances produced by living bodies such as the plants as well as the animals. So just remember, they are generally nitrogenous because they are proteinaceous in nature. Enzymes are also called as biochemical catalysts. Why are they called as biochemical catalysts? Because large number of chemical reactions that occur in the bodies of plants and animals to maintain the life process is catalyzed by these enzymes. So every biological reaction requires different kinds of enzyme, and each catalyzing different reaction. A living cell on the average contains 3000 different kinds of catalysts. Okay, so enzymes are so much vital for the biological processes that without them life process is impossible. Think of the whole digestive system. For this digestive system, different biological enzymes are needed, and these biological enzymes enhance the reaction to a large extent. After that, we have the steps of enzyme catalyst. What are the different steps involved in the catalytic reaction? So here the first is binding of the enzyme to the substrate to form activated complex. Next is the decomposition of the activated complex into the product. Just remember, whenever a catalytic reaction takes place, what actually happens? It is the catalyst that binds with the reactants in the active site of the catalyst. After the catalyst has bounded, then what happens? It forms a complex. Now when the complex is formed, that is called an activated complex. Now this activated complex, on decomposition of the activated complex to form the product, we form different types of product. These are the various steps involved in the enzyme catalysis. Is this part now clear? After that, we will discuss the characteristics of enzyme catalysts. What are the different characteristics of enzyme catalysts? The different characteristics of enzyme catalysts are as follows. They are like this. That is, they are highly efficient. One molecule of enzyme can transfer 106 molecule reactants per minute. They are highly efficient. One molecule of enzyme can transfer 106 molecule of reactants per minute. That is the first important factor. Next is they are highly specific in nature. Example, urease catalyst hydrolyzes the reaction of urea only. So see, try to understand, they are very much specific, means one catalyst is specific for one biological reaction. It is not actually specific for any other reaction. That is, here they have told you that urease will be activated only in this reaction of urea. If I take any other enzyme, what will happen? The reaction will not take place. They are active at optimum temperature. What is the temperature? 298 to 310 Kelvin. That is, the catalyzed reaction becomes maximum at a definite temperature called the optimum temperature. And after that, next point what we have is that they are highly active at a specific pH called the optimum pH. The enzyme activity can be increased in the presence of coenzymes, or which are called as promoters. Now activators are generally metal ions, that is sodium ion, cobalt ion, copper ion. These are enzymes and increase its activity. Next is the influence of inhibitors. Enzymes can also inhibit the presence of certain substance. These are simply the characteristics of enzyme catalysis. These are the characteristics of our enzyme catalysis. Is this part now clear? So before I end in this part of the chapter, we have discussed about, as we start the chapter, we have discussed about different types of catalysis: homogeneous, heterogeneous, activity of a catalyst, selectivity of a catalyst, shape selective catalyst. Then we have discussed about enzymes, types of enzymes. We have discussed characteristics of enzyme catalysis. We have discussed. After that, we have discussed what is called a type of true solution. These are the topics which we have discussed. Okay children, that was all for the day. Thank you.